So I'm talking about some joint work with Hector, Jonathan, and John, funded by the NSF. Okay, so the idea is I'm talking about a new algorithm called TINIC, and what it's going to do is infer the tree of blobs for a network. Okay, so what is the tree of blobs here? So if this is your model network over here on the left, you have cut edges like that, so edges which will subtend a clade leading to the bees, and you're going to have reticulate subgraph structure, which we're going to call a blob. And the tree of blobs derived from that network on the left is that in which all these blobs are contracted to a single node. So down here, if you look at this node, you'll see that corresponds to that blob right there. So cut edges remain cut edges, and reticulation history subgraphs gets contracted to a node. Okay, so TINIC is developed under the network multi-species coalescent model, which models incomplete lineage sorting. The model network is binary. Okay, it has cut edges again, and blobs contracted to nodes. The data input for this algorithm is a sample of gene trees assumed to be generated under the coalescent model. Okay, what we're going to do is infer the tree of blobs. And this is a quick talk, but the real idea here is that you want to sort of look at these high-degree nodes as a place where you want to further investigate possible reticulate evolution in your data. Okay, and the other thing I should say right now is that TINIC is a quartet method. So here we're sort of have the model network on the left and the tree of blobs on the right. And what I'm going to do is I'm going to look sort of 4 taxa at a time. So if I look at C1, C2, C3, and C4, on the true tree of blobs, it looks like a star tree when I focus my attention only on those 4 taxa. And if instead you look over here at A, okay, pick any of the 4 A's, on the tree of blobs as well as in the model network, we have a cut edge, and so it looks like it's related by a quartet tree. Okay. All right, so how do we do this? So I'm back in the model world here where I have a true network over here on the left. And what I'm going to do is classify quartets on the true model network in terms of their topology or their graph-like structure. So if I just focus on A1, A2, A3, A4 over here, and I look at the induced graph on my model parameter, my network N, it looks like a tree over there, a quartet tree. Okay, and we're going to call that a T quartet, T for tree, a tree quartet on N. And the induced graph is a quartet tree. But instead, if I look here like at A1, B, H, and D, and then I look at the induced graph on my model network, you'll see that it looks like a blob or a network. Notice it's not level 1, it's level 2 in this picture here, and that's what it looks like. In the tree of blobs, of course, this blob right here is going to be contracted down to a node like that. Okay, so we call that a B quartet for blob quartet. Okay, so one of the problems for TINIC, or a challenge that we had to overcome, is that you might have a B quartet on the true network N. So this is going to be A, 1, B, C, and D. And the definition is rather technical and mathematical, but the idea is that you have a path leading from each of the 4 taxa entering the blob independently. So there's something here, 4 distinct paths that lead the taxa to the blob. But when you look at the induced quartet graph, okay, you're going to get a tree. So you have a blob quartet on the true network, but when we're looking at quartets in data, they're going to seem to be tree-like in reality. So the point is that quartet data from gene trees is only indirectly going to tell us about the topology of the true network and its B quartets. Okay, so what's the data? Given a sample of M gene trees on N taxa, we're going to, in the empirical setting, we're going to estimate quartet concordance factors from gene trees. So we count up the number of times we see quartet tree AB versus CD, AC versus BD, and AD versus BC. Okay, so the quartet concordance factor, or the empirical one, is the vector of frequencies of quartets displayed on M gene trees. Okay, as a simple example, if you have 30 taxa, 1,000 gene trees, there's roughly 27,000 quartets. You count up how many times you see various quartet trees, 9, 17, 33, 60, and then we're going to normalize to make this a distribution. So our empirical CF will be 0.917, 0.033, and 0.06, like that. Okay, and here's something where the model comes into effect. When you're looking at these quartet CFs, these empirical quartet CFs, the expected values should be PQQ for a T quartet. So you have two entries which are equal, the QQ, and typically or generically less than the third P. And in contrast, when you look at a B quartet in data, so this would be sort of the induced graph is actually a B quartet, you're going to see a three-tuple where all three entries are different, so PQR, like that. Okay, remember, these estimated concordance factors are input for this method. Okay, and I should point out that for these B quartets, or for these T quartets here, PQQ, some of those are going to appear as PQQ quartets in data, but actually on the true network, they're really B quartets. Okay, so step 2, we perform 2 hypothesis tests. So these are hypothesis tests under the network multi-species coalescent model. The first one occurs at test level alpha, and the idea is the null hypothesis is that you have a PQQ. Okay, we have a T quartet in our data, so the empirical CF will look roughly like PQQ format. These hypothesis tests are sample size dependent, okay, so how many gene trees you have depends on whether you accept or reject the null hypothesis. So the cut test for whether or not you have a T quartet or a B quartet like that. And then we have a second test, which I had to speak way too rapidly about, but here we're going to have the null quartet is, CF is of the form PPP. In other words, you don't have enough information in your sample, in your data, to tell whether or not you should resolve the star tree over here into a T quartet or something which might be reticulate-like. Okay. All right, so step 3, what do we do? So, let me back up. Excuse me, went too quickly. Okay, so what we're going to do is you're going to look at your 27,000 empirical concordance factors, your 27,000 quartets, and you're going to go through, and based on these 2 hypothesis test results, you're going to classify every single quartet either as a T quartet or as a B quartet, like that. Okay, so you have this initial list telling you which of the foursomes are B quartets and which are T quartets. Okay, that's not quite enough, so what you have to do is pass over this list again and iteratively use the combinatorial inference rule to get additional B quartets. Because remember, you might have a B quartet on the true network, which looks like a T quartet in your data there. So this requires passing through your list again and again, using a combinatorial rule that pieces together or recognizes additional B quartets based on the list of those who are currently classified as B quartets. So this is very combinatorial in nature. And the point, again, is to reclassify some current T quartets as B quartets like the one I tried to highlight above. Should have been a B quartet or is a B quartet on the true network, but you can't tell when you focus on quartets only. And then the last step is we use a quartet distance introduced by John Rhodes in 2022 to get a pairwise distance table. And once you have this pairwise distance table, you just use your favorite distance method, neighbor joining, FastME, to construct a tree of blobs. I had to take a sip in order to keep up this pace. Okay, so quick theory. Under the network multi-species coalescent model, this Tinnock tree of blobs is a consistent estimator of the true topological tree of blobs. So increase your number of gene trees, and you'll get the true topological tree of blobs. Okay, so how does Tinnock perform in simulation? So here's my model network over here. It's not level 1. You have stacked blobs, B versus C here. You have a tree-like group A, and you have sort of repeated, distinct, or gene flow instances in the C. Okay, so we simulated M gene trees with phyloco simulations. Hang around for talk number 3, given by Cécile here. And for a number of different sample sizes, so 300, 500, 1,000, and 10,000. And to model different levels of ILS, we scaled the branch lengths in this model network by factors of 1, which is what you see, 0.5, and 2. Okay, so how does it perform in practice? Okay, so what you see here for a sample of 1,000 gene trees, when the star tree has been tested not to flag any quartets initially as B quartets. Okay, so using the second test, everything is considered tree-like. And what you see here is entirely typical. So as you decrease the level alpha of the cut test, we're going to get increasing resolution in the tree. In other words, when alpha, the test level is big, you're sort of willing to accept quartets as B quartets, and as you decrease it, you're less eager or less willing to accept that. And what you see, of course, is that, so here's a tree of blobs, which is less resolved, more resolved to the right. Out first pops the A group, and then interesting, out pops this cut edge and also the C blob. Of course, the tree blob continues. And finally, for a long range or a large range of alpha test levels for this cut test, we get the true tree of blobs there on the right. Okay, so as alpha decreases, more resolution, since fewer quartets in that very initial step are flagged as B quartets. Okay, so important takeaways. Some blobs are easier to detect than others. So the A tree popped up first, followed by the C blob. And then the other thing not shown is that the sample size really matters here. Okay, so TINIC gives excellent results over all regimes of parameter space when the number of gene trees is 10,000, 1,000, and 500, but not so true for 3,000. You get the A tree and the C blob, and then it's going, how am I doing? 10? Okay, cool, 2 more minutes. All right. So not only in simulation, but of course we applied this method to real data sets. So in a 2020 paper of Vanderpool et al., there's a data set of primates. And in that paper, there is published this graph, or this network, showing multiple reticulation or hybridization introgression events between the Papionini. This was also studied in a forthcoming paper on finest. Okay, so finest will infer a level 1, so cycle zoning networks, from sequences using a quartet-based method here. Okay, and here you see 1 cycle, down below you see 2 cycles, and clearly there's a difference between the conclusions that these papers draw about these data. So we ran TINIC on them, and interestingly, our analysis is completely in concurrence with the finest one. Okay, so for, I guess, small values of the test on the right, we get this tree, which was sort of the, there's 1 cycle, and on the right, we get 2 cycles, like that. Okay, so TINIC is really, really fast. You do have to infer gene trees first. That's where the time comes in. But once you have your gene trees as input, it's really quick. So for the simulation, we're talking on the order of 37 seconds, and for the Vanderpool data set, 28. Okay, so we're talking under a minute here. Okay, and as a last analysis, we looked at a Hawaiian flowering plant class. And just to show you, there's considerable discrepancy between what Phylonet outputs and what SNAC puts out. Okay, a tree versus a 2-cycle network. And so we ran TINIC also on these data, and we don't agree with either of them. Okay. So if you were to get the tree of blobs for this SNAC network on the right here, it wouldn't agree with what we have, like that. And one thing there is that the data, the gene trees are really poorly resolved. And so that's just something to keep in mind. Okay, I am finished. Woohoo! And here is... There's no time for questions? Okay. Thank you.
So thanks, everybody, for being here. I'm going to talk about 2 things. One is a coalescent model where we add correlated introgression to the traditional coalescent model, and then talk about the simulator phylocal simulations. So I will start with phylogenetic networks, although Elizabeth talked about them. I will give a brief review, and then at the end I will give an example application from all this. This work is joined with John Fogg, who is a graduate student, and Elizabeth Allman here today, who started the symposium. All right, so, and this screenshot is from the documentation for the software. All right, so for some people, I tend to talk about networks as phylogenetic networks. In the population genetics community, many people refer to these networks as admixture graphs, and they really are the same graphs. So they just two names for the same thing. This is an example from more the population genetics literature, a very recent, super interesting paper. There are sometimes estimated, like Elizabeth mentioned, Nanook, SNAC, Phylonet, and from population genetics, they are estimated more often using F statistics. Yes, working. This is an older example. There is just so many of these networks coming out all the time. And this is really cool when we can use ancient DNA to get more complex graphs. So these methods need to use a coalescent model because otherwise ILS can be confounding and we might want to use networks to just explain the discordance that is due to ILS. And the coalescent is a model to model ILS. And something that is rarely talked about is an assumption for graphs about inheritance. So this is on a tree, and just to remind you that the coalescent model on a tree needs to have edge lengths that at least we need to have coalescent units, which are number of generations divided by population size. And this cartoon is just to say that C and D in some gene trees can appear as sister because of ILS. Now, if we're on a network, we have some edges that have, some nodes that have two parent edges because some nodes are hybrids getting genetic material from two parental populations. And I need to have an extra parameter that tells me what proportion of the genome was inherited from one parent and what proportion from the other parent. So then here, CD can be sister either because of ILS or because it was in introgressed genes, just like Dylan was talking about. So what if I have a network, either I sample two individuals from C, or maybe actually C speciated again, and there were multiple species that I sampled below, C1, C2. And what if these two lineages didn't coalesce for some of the genes? Then we need a model here to say how these two lineages choose their parents. Do they choose their parents in the model, coalescent model, independently of each other? Do they choose the same parents? Or do they choose them in between in some correlated fashion? And this model is, this assumption is typically not discussed in the papers that use a multi-species coalescent. So in many papers, like in Phylonet, in SNAC, TreeMix, AdMix tools, there is this assumption that if we have multiple lineages that arrive at a hybrid node, the two lineages choose their parents independently of each other. So if the second lineage here chooses a parent from the right here, that does not affect the choice of the first. That could choose its parent from the left or from the right. So probability of this event would be gamma times gamma and 1 minus gamma times gamma. Okay. So this is the most common multi-species coalescent model. There is another model that is used in other papers, like in GT-Mix, in NetRox, in Finest, so in other papers, where when two lineages come at a hybrid node, they have to choose the same parent. So either both choose this parent with probability gamma, or both choose that parent with probability 1 minus gamma. And I'm not sure in reality what model is most appropriate, okay? But so this assumption could have a big impact. All right. So what we designed is a model that would bridge in between the two extremes. So we have, and what we showed is that a model with correlation parameter rho that would bridge between 0 independent inheritance to rho equals 1 common inheritance, and we have anything in between, that could arise from selection. So selection after gene flow. Take loci that are advantageous coming from the introgressed parent population. Then after selection, if I sample individuals after selection, and then I look at their history back in time, most likely all lineages are going to come from the introgressed parents. If instead I'm looking at other loci that maybe are on the X chromosome and they're selected against the introgressed parent, then maybe for them, the best gamma would be 0 and all lineages would come from the other parents. And so this correlation could come because of selection. We don't know in practice what's the best model, but so what we can offer for now is a simulation tool, and maybe this tool can be used to see what is the impact of this assumption downstream. So this is a Julia package that is part now of the Julia Philo suite. It integrates with other packages in Julia. How you use it, you load the packages, you load your network, you read your species tree, and here is one example species tree I could load. I could plot this using a companion package. And then there we go. We just simulate from the coalescent. I'm giving my species tree. I'm saying I want 2 loci, 3 individuals per species, and let's say inheritance correlation 0, that would mean independent inheritance. And so this is what I might see, so I can just plot all my trees like this. This is another example where I'm asking for very strong correlation. And I'm plotting this example because here you see that in some loci, all the B and C individuals, which were below the hybridization, they all came sister to lineages from A. So they were all inherited from the same parents. And in other loci, they're also all inherited from the same parent, but a different one. So it's just to illustrate the impact of this correlated inheritance on the gene trees. In the package, we also have ways to keep track of how a gene tree is embedded in the network by these extra nodes that have some tags to map them to this, where they came from in the species network. And so using this, then there's a lot that we can do with this package downstream. So for example, we can simulate rate variation. And writing Julia pipelines in Julia is very easy, so we can get a species network that we could simulate from something else. Simulate the gene tree with phylocal simulations, and then use some other seqgens, say, to simulate alignments. So I'm going to go faster, but this is what the code would look like. You assign maybe you simulate some rates for each lineage. So here it's a rate for a lineage in the network. And then I can transform my gene trees using these rates at populations, and then pipe this into seqgen. So that's what the code would look like. Just to show it's actually fairly simple. And thanks to everybody who is part of this effort, funders and co-authors, and yeah, thanks to you for being here. So in this package, there are completely unlinked loci. Yeah. So, yeah.
All right. Thanks, everyone, for coming. I'd like to start by just mentioning that I live and work in Toronto, which has been indigenous land for thousands of years, until about just over 200 years ago. It was essentially stolen by the British, and we are very much in the process, I believe, of reconciling and decolonizing this land. So the main kind of piece that I want to get home today is just that spatial patterns in genetic diversity reflect spatial history, right? So we see this in this example from humans, where individuals that are more similarly genetically in this PCA come from geographically more similar places. So we see isolation by distance. And patterns of relatedness that we see in genetic diversity can help us, make these broad depictions of how species have moved across the landscape to get to where they are today. So here I'm switching to the more interesting organism of Arabidopsis thaliana on the right, expanding out of Africa and westward from the Middle East. And so there's kind of what I think of two main approaches to infer spatial history. One is the kind of non-recombining sequence side of things. We're dealing with a mitochondria or a Y chromosome, where relatedness is described by a single tree, right? And here we're in the realm of phylogeography. There's kind of a way to put an explicit spatial model on this object of relatedness, and that allows us to infer things like where ancestors were and how fast lineages have moved to be where they are today, right? So this is an example of rabies in northeastern US virus. The other realm is kind of where we have recombining sequences. This is a human autosome, for example, or a Arabidopsis autosome. So here we have more information. There are many trees that describe the relatedness of the individuals. But historically, there's been no real good object to model movement on. So this is the realm of population genetics, where we have summary statistics that we look at to kind of infer something about the spatial history, but it's not kind of done in the explicit way that phylogeography is. So we might be left doing something like 23andMe's geographic ancestry analysis, where they'll take your genome, compare it to a bunch of pools of samples from across the world, and say what % of your genome is most similar to, say, the Irish pool that they have, and that'll be your % Irish. But it's quite an imprecise statement to say you have some geographic ancestry from this political boundary, but we don't know exactly when that was. And that's kind of the cost of having this kind of implicit model of movement. So I think what's exciting is that we have new possibilities now that we can infer these things called ancestral recombination graphs. Right, so as I just mentioned, in a recombining genome, the relatedness between individuals is described not by a single tree, but by many trees. And so you can think of moving along the genome. This part of the genome is described by a particular tree, but a recombination event happens. Now we have a new tree that describes the relatedness between individuals, and so on. We have this sequence of trees, this tree sequence. And the reason I'm in this network symposium is because you can weave together all these trees into what's called the ancestral recombination graph, where we not only have these coalescent nodes as we typically think about, but we also have these recombination nodes where the lineage history splits and part of the genome goes to the left, part of the genome goes to the right. And because of these recombination nodes, we get these loops in these ancestral recombination graphs, which is something that we have to deal with when we're going to do some spatial inference. So the exciting thing is that we can now infer these args, or tree sequences, for very large samples across the whole genome, and they provide an object to model movement on. Okay, and what I'm going to talk to you about today are two approaches that we've developed to do just this. So the first is the non-networky part of the talk, and I'm going to try to go through this relatively quickly to get to the network stuff. But I've been working on this non-network part for a long time, and I think it's pretty cool, and it's ready to apply to data, so I want to share it all with you. The step one is that we kind of do the simplest thing possible here. We take our sequence of trees, we sample trees kind of sparsely across the genome so that the trees are relatively independent from one another, and that allows us to then apply our Brownian motion model independently at each of our sampled trees. And applying the Brownian motion model is just like a phylogeography. The sample locations are just normally distributed around the root with some covariance that depends on the shared times of them. So if two lineages share more time with each other, then their spatial locations covary more. They have less time to split apart from one another. So the key object is really the shared times matrix, S, which is what we can get from the tree sequences. Once we infer the tree sequences, we know S, and then we can infer mu and sigma. And so in step 3 of this process, we first estimate the dispersal rate. So we multiply the likelihoods across our sparsely sampled trees, get a maximum composite likelihood estimate of the dispersal rate using the entire genome. And step 4 is to locate ancestors, say, of sample 1, 10,000 years ago. At every sampled tree, we can ask that same question. Where was samples 1's ancestor at that time? We get thousands of ancestral locations for sample 1, thousands of distinct ancestral locations for sample 1, and we can visualize a cloud of ancestry, a distribution of ancestry for that sample on a map. So I'm going to skip over the simulation bit of this project and get right to the fun stuff of applying it. We did this for Arabidopsis thaliana because it has great genomic resources, wide geographic range, very interesting spatial history. For our purposes, the main piece we need to remember is that there's kind of this Middle Eastern population and this North African population that expanded and met each other in the Iberian Peninsula here in Spain and created a bunch of admixed individuals. So this is kind of the piece that we'll look at briefly. And so the data set we used was 66 high-quality long-read genomes where we could infer the tree sequence with quite a bit of confidence. And it was sampled in a very smart way, where we have these, even though we don't have a ton of samples, we have these orange samples that are from the North African lineage. We have these blue samples that are primarily from the Middle Eastern lineage. And then we have green samples out here that are much more deeply diverged from the rest. But this provides us enough variation to do some interesting things. So the first step was to infer the dispersal rate. I don't really want to get into the details too much, but we inferred a dispersal rate of roughly 50 kilometers per generation, which is a large number if you compare it to how far a seed falls from a plant, but is a small number if you believe this picture of spread from, say, the Middle East to the UK in about 10,000 years or something. So we think we're striking some balance between small-scale, short-term dispersal and the amount of dispersal that's required for these longer time frame, larger-scale movements, at least under a model of Brownian motion. And then what I think is the fun part is applying this to ancestor locations, and so I'll show you just one picture of that, where we compare a non-relic sample. This is from the Middle Eastern refugia that is now in northern Sweden, and a non-Iberian relic from the Madeiran Islands. So this is a more deeply diverged sample here in the bottom. And this is the distribution that you see as this contour plot of where its ancestors are about 1,000 loci, if I remember correctly. And the interesting bit as we move back in time is that we see this Madeiran sample stays pretty close out there, doesn't kind of move very far in, and this is consistent with the island being colonized 20,000 or 30,000 years ago. I can't remember exactly, but it's fairly deeply diverged, and we picked that up. These ancestors are still are far from the continent. And what the Swedish sample is probably reading a little bit too far into it, perhaps, but there's kind of two peaks of this distribution. One that's maybe consistent with a post-glacial expansion is further south, and then there's a further north cluster, which could indicate kind of multiple bouts of colonization of Sweden, which others have hypothesized before us. So we'd have to do a little bit more work to kind of say that more confidently, but this is the kind of things you can do with this method is kind of make some visuals and see how it's consistent with different hypotheses that people have. So this method was, we had a preprint about 3 years ago, and then 2 days ago I got a revised version up that I'm very happy about. So check it out if you're interested. The cool thing about it is ready to apply to large data sets. We have relatively efficient code up and ready to go, and I'm very keen to help anyone work on a data set. So let me know if you have trouble using it, or if you just use it and want to tell me about it. I'd love to hear. The downside of this approach, to kind of lead into the next part of the talk, is that it throws away most trees. And so what we would rather do is work with the entire arc rather than splitting it up into trees and sparsely sampling them. But what we wanted to do is basically just extend the Brownian motion model to this graph object The issue is that we now need Brownian motions to kind of split here at node 23, but come together back here at node 14, meaning that those lineages are no longer independent. It's a more complicated model of Brownian motion, and we didn't know how to do that. Because everything is Brownian, we know that the sample locations will be multivariate normal. The likelihood will be multivariate normal, but we don't know what the covariance will be between those locations. My first thought was, well, let's just do the same thing as the trees. We'll just, instead of a single path from like 2 back to 5, there'll now be multiple paths per sample. So one has 2 different paths back to 5 in this example. And we can just treat each path as like a sample lineage, and we'll make this bigger matrix of shared times and just do all the same stuff we did before. But I wasn't sure that that was the right thing to do, so I tried to kind of extend from basic principles what the correct covariance matrix should be for more complicated args, and I couldn't figure it out. But fortunately for me, Puneeth Daraje, who's here in the front, just joined the lab, and I asked him if he had any thoughts, and within a couple weeks he had derived the covariance the principled way, and also proved that it was the same as the heuristic that I had done before with some corrections. So I was very thankful that Puneeth was in the lab and that I had asked him the question. Because now we had the full likelihood on the full arg to estimate dispersal and locate ancestors. And so everything seemed great. We teamed up with James Kitchens in Graham Coop's lab to develop efficient algorithm. So, these args get pretty big and unwieldy, and calculating the shared times of all lineages becomes a bit of a mess. But Puneeth and James figured out some very clever ways to get this down from, hundreds of thousands of seconds to less than a second by doing things in a smart way. So we have it all figured out, but then we attempted to apply it to dispersal rate. And this blue line at the bottom shows the likelihood method, the tree method that I just showed. So the composite likelihood asymptotes as you include more and more trees along the genome. The black line shows what we get from our arg, which is it was a horrible discovery to see the dispersal estimate just increase and increase as we included more genome. In particular, as we include more recombination events, we think. And so the issue here is that we have the wrong model of how the spatial process works. We're forcing these Brownian motions to meet precisely, randomly, at a given recombination node. And that's a very unlikely event. And people have known this. This is why more complicated spatial coalescent methods exist that Barton and Etheridge and others have worked on. But this is a model misspecification. And if you force these Brownian motions to meet a recombination node, it causes all sorts of artifacts above that to make that event more likely. And so that's why we're getting this increased dispersal estimate as we include more recombination events. So I'll do this part quickly. When we turn to locating genetic ancestors, we see some benefits. There's less uncertainty because we're using more information. We can visualize recombination events, so we can trace a lineage back and actually know when it splits and kind of visualize this ancestry from multiple founding populations, perhaps, which was a benefit. But this bias, this excess constraint problem sneaks back in, and we actually have a bias in where we locate ancestors. So this is starting with a single tree, and as we use more and more of the genome moving down, our estimate gets worse and worse. We get more and more error because we have a model misspecification error. And so a promising approach turns out to be a little tricky. The good is that we have a full likelihood on the full arg with an explicit spatial model. The bad is that there's a pathological dispersal rate and biased ancestor locations. And so what I want the take home to be is that it's not clear how to extend a very tractable, simple, conceptually nice model of Brownian motion from trees to graphs. More generally, because I'm running out of time, these args contain a lot of information, and one exciting direction is develop more methods to extract the information, but it's not always going to be straightforward, I think, to extend methods from these trees to graphs, but hopefully there is a way. With that, I'd like to thank Puneeth and James especially, and as well as Graham. Thank you.